international school. Before we start, I want to ask you all a question. Did you know that de-extinction is actually possible nowadays? However, not all extinct species could be brought back, as some recent extinctions are caused by human activity. We could give them a second chance by bringing them back. Let's first talk about the benefits of de-extinction and why scientists should use de-extinction. So why use de-extinction? Why spend so much time on something so expensive and might not even succeed? The thing is, over 1 million animal and plant species are endangered. Since 1900, over 40% of amphibians, 33% of reef-forming corals, and over 1/3 of all marine mammals are at risk. On average, over 30,000 species disappear every year, and the World Animal Foundation predicts that up to 50% of all animals will die off by 2050. So the first benefit of de-extinction is that de-extinction helps restore species that shaped entire ecosystem when alive. For example, the mega herbivore, the auroch. The auroch maintained open grasslands through grazing, prevented forests from overgrowing, and created habitats for birds, mammals, and insects. Also, the pursuit of de-extinction encourages innovation in genetics, reproductive biology, and conservation science. CRISPR and gene editing techniques are improved for uses far beyond resurrection. Also, cloning improves success rates for endangered species conservation. Now, let's talk about the 3 specific ways to de-extinct. The first method is backbreeding. Backbreeding is the process of breeding living relatives to resemble the extinct species. For example, the quagga, a subspecies of the zebras. The last female quagga went extinct in 1883 at the Chura Magissa Zoo because of excessive hunting. And to resurrect the quagga, scientists first had to select similar-looking zebras and breed them repetitively until the last animal looks extremely like a quagga. However, this method is very time-consuming and only works on animals with very close relatives. The second method is cloning. Cloning is the process of making an exact copy of the living organism. And to clone, scientists first extract a preserved nucleus containing DNA from an extinct species body cell, then inject it into a nucleus cell of the living relative. The embryo will develop and implant the embryo into a surrogate mother. An example of the extinct species that will use this method to get resurrected is the Pyrenean ibex, also known as the bucardo. The last female Pyrenean ibex, Celia, died in 2000. And to resurrect the bucardo, scientists first used the frozen repair cells that is preserved in liquid nitrogen for somatic cell nuclear transfer. And then 407 embryos were developed. Half of them were transferred to 57 goat mothers, but only 7 pregnancies resulted, and only 1 baby ibex was born. However, the baby ibex died almost immediately, making it the only animal to go extinct twice. And the drawbacks of this method is that DNA decays after 1,042 years, and it has lower than 5% of success rate, and the animals usually have health defects. The last method is a combination of CRISPR and cloning. CRISPR stands for clustered regularly interspaced short palindromic repeats, and it is a powerful gene editing tool that normally works with Cas9, an enzyme that cuts DNA molecules by chemically breaking the bond, holding the DNA backbone together. So CRISPR, the combination of CRISPR and cloning, first replaces the living relative's DNA with the extinct species' DNA. And to do that, cloning extracts DNA from the extinct species. Then CRISPR is used for the extracted DNA to replace the matching DNA sequences of the living relative, thus creating a full new set of DNAs. So parts of the DNA are cut out, then the cell's natural repair system either disables the gene or inserts new genetic material. An example of extinct species that will use this method to get resurrected is the woolly mammoth, and it is predicted that it will be revived by 2027. However, de-extinction is not perfect. It has drawbacks. Changed habitats, such as if the mammoth is put into the current Arctic, which is a shrubland right now, it may decrease infiltration, but may also damage existing ecosystems there. Also, resurrected species may turn invasive. For example, dire wolves. Dire wolves are more powerful and stronger than the current wolf species and may overpower them. And as resurrected species have low genetic diversity, it makes them prone to illnesses and spread diseases. Lastly, resurrection has very huge costs, and the money will be better used for the current endangered species than to bring back died out ones. And new creatures are hybrid. They could be over 95% of the extinct species, but really 100%. In conclusion, de-extinction is not the top choice right now. The priority is to protect existing endangered species, as it costs less and only requires your willingness to help. The value right now is that it's a good scientific research project and a good backup solution. For now, we should just save wildlife first. Resurrection isn't fully reliable. Thank you.